Good morning, good afternoon, or good evening, wherever you are joining us from across the globe. It's a genuine privilege to welcome you virtually to Gene Solutions Industry Symposium at the 30th International Conference of Prenatal Diagnosis and Therapy, ISPD 2026. My name is Ida Deleskog Lindstroem, and I'm the Global Medical Affairs Director at Gene Solutions. This marks an important milestone for us, our first major international symposium dedicated to NIPT. The theme, Advancing Prenatal Screening, Validation and Clinical Utility of Integrated Non-Invasive Testing with focus on carrier screening and single gene disorders, comes at the pivotal moment. While NIPT has transformed screening for common chromosomal aneuploidies, significant unmet needs remain, particularly for single gene disorders and carrier screening in diverse populations and for broader pregnancy risk assessment. Today's symposium brings together leading experts to examine validation, real-world utility, and forward-looking innovations in non-invasive testing. Before we begin the scientific talks, I would like to briefly introduce Gene Solutions and the journey that has brought us here. We are proud to share our journey from Asia to global leadership in prenatal care. Co-founded in 2017 by two molecular PhDs from leading universities in the US and one medical doctor, our founders brought advanced expertise home. With a shared vision to make genetic testing more accessible, clinically meaningful, scalable, to improve outcomes for all. Now expanding globally with innovations in precision oncology, multi-omic liquid biopsy, they have built this mission from their home country. In 2018, we developed and validated TriSure NIPT, later incorporating advanced AI and machine learning to support carrier screening integration in 2022, with expansion to dominant single gene disorders in 2023. These advances reflect on our commitment to scientific rigor and are supported by more than 20 peer-reviewed publications in NIPT and in genetic studies. To date, TriSure has supported more than 2 million pregnant women across over 5,000 hospitals and clinics in Asia alone, with expanding operations to Europe, Middle East, Africa, and Latin America, backed by our global network of 17 next-generation sequencing laboratories. Today, we are honored to support ISPD's 30th anniversary as a premium partner. With that, I'm pleased to introduce our distinguished experts who will provide complementary perspectives on prenatal screening, spanning laboratory rigor, clinical implementation, regional insights, and data-driven innovation. We are privileged to be joined by Dr. Natalie Chandler, Principal Clinical Scientist at the Rare Disease Inherited Laboratory, NHS North Thames Genomic Laboratory Hub at Great Ormond Street Hospital in London, UK. Dr. Chandler will present from trisomies to broader prenatal genomics, evidence, challenges, and a framework for responsible clinical integration. Next, Dr. Sang Hung Tang, Medical Director, Medical Genetics Institute of Ho Chi Minh City, Vietnam, will share on large-scale real-world evidence of expanded carrier screening via routine NIPT from Asian pregnant women. Finally, Dr. Hoa Giang, co-founder and Chief Data Science Officer of Gene Solutions, based in the US, together with Dr. Min Ju Phan, our Head of Data Science based in Australia, will present on multi-omic biomarkers and AI for early risk stratification of gestational diabetes and preterm birth. Following the presentations, we will conclude with a panel discussion and a Q&A. I warmly encourage your active participation. Thank you once again for joining us today. And it's now my my pleasure to invite Dr. Natalie Chandler to begin.
Thank you for the opportunity to speak today. Firstly, for my background, I am from the North Thames Laboratory in London. I am a clinical scientist and I have been involved in setting up and running the non-invasive prenatal diagnosis for monogenic or single gene disorders in the English NHS Genomic Medicine Service. This is my area of expertise, so the content on my talk is focused on monogenic disorders. So all of our services are considered diagnostic, and this is because all testing for high-risk pregnancies is based on family history or an ultrasound anomaly. We have validated our assays to detect each bespoke variant, or for recessive conditions, the haplotype-based method that we use, which is variant agnostic, has been validated on many cases, and therefore we're able to say that no invasive confirmation is required. So for the rest of the talk, I will be talking about non-invasive prenatal screening, which is where you need invasive testing to confirm the result. So all of these tests involve testing of cell-free DNA from the maternal plasma, and this contains a mixture of the maternal cell-free DNA and the fetal cell-free DNA. There's been lots of studies that have shown there's very high analytical validity for de novo and paternal variants, and therefore I'm not going to go into this in any detail. However, there is still areas that need to be addressed and reduce sensitivity for maternally inherited variants. So as we've shown that analytical validity is slowly getting there, we now need to be asking what we should be screening for, who we should be screening, and will this increase invasive testing rates? Can we avoid false positives? What do we do about maternal somatic mosaicism? Issues with variant classification? Will the variant cause disease? And also, what about false negatives? What do we need to consider in complete coverage? Are genes fully covered? Is there a little dropout? What types of variants are we testing for? So I tried to do my best to actually identify what testing is actually available in the area. So looked at de novo X-linked paternally inherited variants, autosomal recessive testing, maternal carrier screening, and X-linked inherited. There is large variability on the testing that is available out there and the gene content on the panel. However, to note, none are offering X-linked inherited conditions other than fetal sexing, from what I understood. This was actually really challenging to do, and it's actually quite hard to get the information. And what is notable is that there is no commercial offering is the same. There's different genes being tested, and this makes it extremely challenging for users to compare. Who should we test? So all providers recommend testing to include the following indications. So patients who want as much information as possible. And on this slide, I have color coded. So these are ones we term like the all risk or the low risk or screening indications. Or in red, we have those high risk pregnancies. So for those offering the de novo X-linked or AD or paternally inherited variants, they all target ultrasound anomalies, including short long bones and increased nuchal translucency. They are recommending testing for those that have a family history of a variant in a gene on the panel, excluding those with maternal inheritance. And also commonly test is recommended for couples with a male partner who is over 40 years old due to advanced paternal age. Now, you don't need to read the slide in detail, but one thing to notice that one of the commercial offerers does have detection rates on their website, and it's just something to be aware of, because although the detection rates for one provider are quoted at 91.96, for most genes, there are some where it's only 47, 53, 78. So just because a gene on the panel doesn't mean you're detecting all the variants. Are they only testing for single nucleotide variants? Are they testing for copy number variants? How much is this a disorder caused by copy number variants? This all comes into it, and this needs to be clear in the counselling and on the reports. So how do the commercial offerings compare with invasive sequencing in terms of diagnostic yield for different clinical indications? And this study by Zhang et al. In 2024, they published the diagnostic yield per indication for monogenic conditions. And in this slide, we compare it to the invasive sequencing yield from the recent systematic review of single gene disorders. from invasive testing. And as we can see, even though the diagnostic yield for skeletal variants using the non-invasive prenatal sequencing assay was quite high, it was still lower than that of invasive. So this means for fetuses with scan anomalies, these results could provide false reassurance, and it's really important that patients are being appropriately counselled based on these results. And it's not always available if these companies are doing direct consumer testing. But however, there are really good clinical applications for non-invasive prenatal sequencing despite these limitations. So, for example, fetuses where you've got scan anomalies and there are challenges in taking an invasive sample, it might be a quicker test to do rather than waiting to have another go. Fetuses with scan anomalies where parents do not want invasive testing. For paternally inherited or de novo variants, you can actually test at an earlier gestation than you can invasively. You can test from, some say, 8-9 weeks gestation. It is a really good option for de novo variants when they're on the panel because there's a low risk of recurrence, and people will say that this might be lower than the risk of taking invasive. Very debatable, but it could be argued. And if scan anomalies are highly indicative of a particular disorder and this is on the panel, then potentially you could say that this test is actually fully diagnostic given the phenotype. Things are moving on though, and obviously at the moment what we have is panels of genes. However, there have been multiple studies in recent years on non-invasive exome and genome study, and this technology is really rapidly developing, and it's showing that across the exome, across the genome, they're working on CNVs. There's been work on UPD. There's been work on all sorts of different variants. So the analytical validity is actually starting to come closer to that of invasive testing, with the caveat of the maternal variants needing some development. So we are starting to approach a time where the testing for most high-risk pregnancies of scan anomalies or family histories could be non-invasive. So what are the issues we need to address to be able to do this? Well, the first is maternal somatic mosaicism, which we do need to consider. So low-level parental somatic mosaicism is usually missed by conventional testing methods. So if a previous fetus has been diagnosed using genome or exome, the mosaicism in the parents might be missed. And all of the methods we are using for non-invasive prenatal screening have to have much higher sensitivity as we are looking for low-level variants. And in our non-invasive prenatal diagnosis service, we've seen parental somatic mosaicism is detected in around 7.6% of families, and this has been replicated by a study from France. So if it's paternal somatic mosaicism, this actually, doesn't impact on the non-invasive prenatal testing at all. It only impacts on recurrence lists. However, if you've got maternal somatic mosaicism, you have potential to cause false positive results due to the variant background in the genomic DNA. So therefore, it is essential that we are either running the maternal genomic DNA alongside the cell-free DNA for all non-invasive tests, or there needs to be a reflex test if a variant is found to check if it's been maternally inherited. So the next thing to consider is variant classification. So we're all comfortable with Richards and Riggs for SMV and CMV classification. However, since these publications in the around 2015 and onwards, there have since been a mass of different variant classification guidelines, so much that it is impossible to keep up and you are unable to reference on a report exactly which guidelines you're using. For example, us in the UK, we use the Association of Clinical Genomic Science ones. I'm very aware that ACMG guidelines are in the process of being updated, and this issue is likely to be addressed, but at the moment, this is what we have. What does this mean in practice? So these are bubble plots that have been shared with me kindly from the GenQA EQA company. The bubble plots, essentially what happens in this EQA scheme is they send around variants to all of the labs that sign up for the scheme and ask them to classify the variant and provide all their criteria. The green bubble shows the expected classification criteria. The red is the not expected and the yellow is kind of deemed acceptable. But what we see in this data is a variety in the classifications, including some from pathogenic to benign. And this is extremely very in particular for the CMV classification. So just to highlight, there are challenges with variant classification. It's still not consistent across laboratories despite guidelines being in place, and we need to do further work in this area. So talking about external quality assurance, this is actually a requirement for accreditation of a laboratory. However, for non-invasive prenatal tests, it is actually really challenging to generate samples for external quality assurance. So for NIPT, for aneuploidy, they used to use, and fetal sex determination, they used to use our rapid biobank. However, this wasn't sustainable, so now they're using manufactured samples using Serocare. There's no schemes yet available for monogenic disorders, and this needs to be resolved. So what schemes are out there? So there are extraction ones out there. There are testing, non-invasive testing for aneuploidies, common microdeletions, fetal sexing. There are variant classification schemes. There are multidisciplinary teams working. So there are various schemes out there, but there are currently no EQA schemes for non-invasive prenatal screening for monogenic disorders. How do we get around this in England? So because we've got the 2 accredited labs, what we do is a sample swap to cover requirements for accreditation. So what happens when we start testing non-anomalous fetuses or fetuses without scan anomalies? Well, if a variant is classified as pathogenic, and we're happy with that, will it actually cause disease? Well, actually, in a lot of recent studies from population biobanks, there's reports of low penetrance. So patients, individuals with pathogenic variants in genes that do not have the features of the conditions. I've picked out key ones here, and I just like to highlight osteogenesis imperfecta and tuberous sclerosis because all of the dominant de novo NIPS panels contain these genes, but there's also recessive genes and many more. And it's really complicated because it's likely to be variant specific. There's environmental polygenic and alternative splicing that is going to impact on this penetrance. And actually, it's really challenging for us to determine the risk for an individual with our current knowledge, and we need to do a lot more work in this area. So actually, in the absence of a family history of a demonstrable phenotype, the likelihood that a fetus with a pathogenic is affected remains low. So how does routine cell-free DNA screening look in practice? So in this study on non-invasive prenatal sequencing 30 gene panel, they tested 2,745, and we saw a lot of the same issues I have previously gone through in this presentation, such as issues with variant classification, false positives due to maternally inherited variants, issues with variable expressivity, variants that were reported that do not fit a dominant mechanism for disease, and also reporting finding of unaffected parents and the penetrance issues where the parents had mild features but unaware they had the disorder. And actually in this study, 78.6 of the variants they've reported were actually from unaffected parent de novos, and actually only 2 of them with de novo. On the next study, which is actually on invasive testing for exomes, where they were testing non-anomalous fetuses, they did 1,766 cases and had 3-year follow-up data on 17, although note this is telephone and not medical. They only had 2 cases that had complete phenotypic fit and 2 with partial. So actually 17.6.47% had no feature of the disorder at 3 years. And also to note that 8 out of 13 were novel truncating variants, and this was the classification was solely based on computer prediction. So just highlighting some quotes from the discussion from this study because they really highlight what we need to address as a society. So they are in agreement that the presence of a pathogenic variant does not equate to a clinical diagnosis. They believe that there needs to be a well-established gene panel that is limited to genes associated with diseases that have severe outcomes. In the absence of prenatal phenotypic features, we need to develop as more stringent variant classification guidelines. And given all of the complexities that I've covered in this presentation, we need to have discussion on how we establish a rigorous analytical framework and how we are able to counsel the patients accessing these tests. Just to summarize, for families with known pathogenic variants, we are approaching a time when many families are eligible for prenatal diagnosis using a non-invasive prenatal diagnosis or non-invasive prenatal screening. Current non-invasive prenatal screening offerings are highly variable in their testing content, and currently For those with scan anomalies, they have a lower diagnostic yield in compared to methods used for invasive sequencing. Variant classification is a challenge. It is not as consistent across laboratories, despite having guidelines in place, and further standardization is required. Laboratories need to participate in external quality assessment for accreditation. However, we really need to develop a non-invasive prenatal screening one for monogenic disorders. Testing non-anomalous pregnancies, we have real issues with variable expressivity and penetrance, and we need to work together to address these. And it's essential that there is counselling in place with genetics expertise for any patients accessing these tests, as they're not always available in direct consumer testing. So these are just my thoughts on how we address some of these challenges, and as a society, what we need to be working towards. So we need to be doing social scientist studies to understand views of healthcare professionals and patients on what we should be screening for. We need guidelines on what to screen for and how to report. We need consensus and guidelines on how to classify variants, particularly in the absence of a phenotype. We need studies on penetrance in the general population, and also sharing of data and larger population data sets so we can start to understand the penetrance. These issues are not unique to prenatal diagnosis. It's also the newborn screening community is also impacted by these same challenges, but it's just more complex for prenatally due to the ethical challenges. And finally, I'd just like to acknowledge all of the funders and patient groups that are involved in all of our groups' research.
Greetings, all ladies and gentlemen. I'm Dr. Sang from Medical Genetics Institute of Ho Chi Minh City, Vietnam. It's my honor to present our work, large-scale real-world evidence of carrier screening via routine NIPT from Asian pregnant women. Here is my agenda for today. I will begin with the clinical needs and our expanded NIPT to include the screening for the most common recessive and dominant monogenic disorder. Then I will focus for the integration of the carrier screening for the most common 18 recessive disease with our real-world evidence from more than 225,000 pregnant women across Asia. The goal of prenatal screening is to inform pregnant women and couples about the risk for the birth defect and provide them informed choices on how to manage the risk. As we can see here, 55% of the birth defects come from genetic burden, and monogenic disorder contribute for nearly half of unidentified genetic causes, making them an important target for prenatal screening. This highlights the need to expand prenatal screening beyond chromosome abnormality to include common monogenic disorder. Traditional NIPT focus only on detecting chromosome abnormality. Our innovation to expand NIPT into two directions, maternal carrier screening and the NIPT for common dominant monogenic disorder of 30 high penetrance genes, with the clinical utility has been demonstrated by Dr. Chandler in previous presentation. Today, I will focus on maternal carrier screening component and our real-world evidence of integrating it into routine NIPT. So we're integrating carrier screening into routine NIPT. Carrier screening is the most effective strategy to prevent for severe and common recessive disease. Professional guidelines now recommend carrier screening should be offered for all pregnant women and those planning for pregnancy. However, clinical burden is still substantial. This is the clinical case that we could not forget or tolerate. A healthy couple with no family history lost their twin daughter within 2 months after birth because of respiratory distress and the cardiomyopathy with unclear diagnosis. Hoping for different outcome, they later conceived through IVF the third try, but their son developed and died soon with the same condition. Genetic testing finally confirmed barren for the carrier of one common recessive metabolic disorder. So this case highlights an important reality. Most carrier couples completely healthy and unaware of their reproductive risk until the birth of an affected child. Together, implementation of carrier screening remains challenging because the limited access to the technology, the cost of the testing, and the challenge in the clinical implementation. By integrating carrier screening into the existing NIPT workflow, we can overcome many of these barriers by simplifying testing, improve accessibility, and early identify at-risk couple during pregnancy. Our solution is to combine both testing from a single maternal blood sample during NIPT after cell-free detection. The NIPT and carrier screen were performed simultaneously from a maternal cell-free DNA. This approach requires no additional blood draw, minimize additional costs, and support for the early detection in the first trimester and the scalable implementation. Its maternal origin contributes for around 90% of the cell-free DNA in plasma. We have developed a machine learning algorithm to detect for maternal carrier status directly from the plasma cell-free DNA. This method has been analytically and linearly validated for both alpha and beta thalassemia diseases. 18 common recessive diseases were selected based on 3 criteria: the carrier frequency, the severe clinical impact, and the clinical actionability. These criteria were consistent with the ACMG recommendation for universal carrier screening, and the carrier frequency were informed by large-scale genomic study in Vietnamese population. Here the detail about the common 18 recessive disease with the associated 19 gene and the major clinical outcome. The panel include alpha and beta thalassemia, leading to fetal high blood and severe anemia, the cystic fibrosis and the autosomal recessive polycystic kidney disorder, leading to multi-system and organ dysfunction, the 5-alpha reductase deficiency, leading to infertility and the disorder of sex development for male fetus, 7 metabolic disorder, leading to metabolic crisis, seizure, organ dysfunction, or even the neonatal death, Pendra syndrome, leading to the hearing loss and the speech and the language delay, the Charcot-Marie-Tooth disease, one of the neuromuscular disorder, leading to the severe hypotonia at birth and early infant mortality, and finally, for X-linked disorder, GCPD deficiency, leading to hemolytic anemia, hemophilia related to severe bleeding, X-linked adrenoleukodystrophy, leading to severe childhood progressive neurodegeneration, and finally, ornithine transcarbamylase deficiency, leading to the neonatal crisis, encephalopathy, and the neonatal death if untreated. For the technology, we use the gap PCR to detect for the common deletion on the alpha thalassemia gene and the exome-based capture sequencing to examine the point and indel mutation in the targeted gene, enabling the detection of more than 7,500 pathogenic deletion and mutation on the 19 gene. Due to the technology limitation, we do not detect the intron inversion of the FA gene, and we could not detect the common recessive disease in the region such as the spinal muscular atrophy, the congenital adrenal hyperplasia, and the Prazoid syndrome recommended by the ACMG guideline. To evaluate for the feasibility and the real-world implementation of carrier screening integrated into routine NIPT, we conducted a real-world study from more than 225,000 pregnant women across Asia, with majority from Vietnam and the rest from Taiwan, India, Malaysia, Philippines, and Indonesia within 1 year period. We assessed for the carrier frequency, the disease distribution, the partner screening uptake, the diagnostic follow-up, and finally the detection of affected pregnancy and newborn. This was a retrospective real-world cohort study conducted from February 2025 to February 2026, including 225,092 pregnant women. Most of the participants were simultaneous pregnancy, with the mean gestational age at 11.4 weeks, representing for routine first trimester prenatal care. As mentioned previously, just one single maternal blood sample. We perform NIPT and carrier screening together based on the plasma cell-free DNA. For the carrier screening, we combine gap PCR and the exome-based capture sequencing based on validated machine learning algorithm with high accuracy for the detection of 18 autosomal and X-linked recessive disorder aligned with the ACMG guidance. Women identified as carrier receive genetic counseling and follow-up with the further testing. At-risk couple then offer prenatal or postnatal diagnosis based on our patient support program. This integration workflow allows a comprehensive prenatal genetic screening without requiring additional blood draw and enhancing cost-effective. Now, I would like to present the key findings of this study. First, we found that more than 1 in 4 pregnant women carry at least one recessive condition. In total, 61,114 women identified as carrier. Among them, 82.4% carry one condition, while 15.6% carry two conditions, and 2% carry three or more, highlighting the complexity of the carrier status in this population. Looking at disease distribution, alpha thalassemia was the most common recessive disease, with the carrier frequency of 8.2%, or 1 in 12 pregnant women, followed by cystic fibrosis of 6.3%, beta thalassemia of 4.5%, and 5-alpha reductase deficiency of 3.4%. Several metabolic disorders, including Wilson disease, phenylketonuria, and C-gene deficiency, also show high relatively carrier frequency. More importantly, 11 of 14 autosomal recessive disease met or exceeded ACMG tier 3 threshold, supporting the inclusion in routine carrier screening. The most dominant disease is consistent with established knowledge about the regional burden of recessive disease, including alpha and beta thalassemia. We also observe emerging findings of the high carrier frequency of cystic fibrosis in Southeast Asia population and 5-alpha reductase deficiency in Vietnamese population. Among X-linked disorders, CCPD deficiency saw a remarkably high carrier frequency of 1 in 20. Although other three disorders were much less frequent, they are associated with severe neonatal or early childhood outcome, defining supporting for the inclusion of the clinically significant X-linked disorder into routine carrier screening in line with ACMG recommendation. Beyond the frequency, we also evaluated the clinical implementation of this integrated strategy. Among 61,114 identified carriers, 27% of partners underwent carrier testing. This identified 1,195 at-risk couples, of whom 229 proceeded to diagnostic testing through our patient support program. Ultimately, we identified 67 affected cases, including 54 affected pregnancy diagnosed prenatally and 13 affected newborn diagnosed after birth. This included clinically significant disorder, alpha and beta thalassemia, Pompe disease, phenylketonuria, Penrose syndrome, and Wilson disease. To conclude our study so that routine NIPT can be expanded beyond aneuploidy screening to provide a scalable carrier screening for Asian population. In over 225,000 pregnant women, this integrated approach identified a high burden of autosomal and X-linked disorder and enable the detection of affected pregnancy and newborn through structured follow-up. As expected, thalassemia and metabolic disorder were the common disease, while high carrier frequency of cystic fibrosis and 5-alpha reductase deficiency need further investigation. The integration of the carrier screening into routine NIPT support informed reproductive decision making, strengthened population genomic screening, and had the potential to reduce the severe inherited disease in Asia population. Future efforts should focus on enhancing partner follow-up, optimizing the follow-up pathway, and enable the implementation of the carrier screening into routine prenatal care. To end my presentation, we have demonstrated to you that our innovation align with the ISPD mission. By advancing the science and the practice of genetic and fetal care worldwide, we will promote the health of children, their mother, and their family. Thank you for your attention.
Hello, everyone. It is my honor to present to you our latest work regarding early predictions of preterm birth and GDM using AI and multi-omics biomarkers. Over the last decade, maternal plasma cell-free DNA has fundamentally redefined the standard of care in prenatal screening. Maternal plasma contains fragments of cell-free DNA released not only from the maternal tissues or from the but also from the fetus and placenta. By extracting the cell-free DNA, we can get a glimpse into the physiological and pathological state of both the mother and the placenta or the fetus. Within a single blood draw, we can capture multiple biomarkers as listed here. However, in practice, genomic and fragmentomics are most commonly used because they are based on cell-free DNA, which is more stable, and both of them can be captured using the same wet lab protocol. So in this talk, I will focus mostly on biomarker derived from cell-free DNA, especially fragmentomics. So the fragmentome of cell-free DNA includes all the characteristics or features of the cell-free DNA fragments. Most notable features are listed here. Fragment length profiling, or FLEN, looks at the shift in the short fragments compared to the long fragments. Because the short fragments often release from the placenta or the fetus. The end motif most commonly used is the former or the 4 base pair at the 5' end of the fragment. This motif can reflect the DNA cleavage profile that changed with pregnancy pathology. Nucleosome footprinting is the spacing of nucleosome along the DNA molecule of the genomes. This can be indirectly reconstructed by looking at how much region is protected, therefore release fewer cell-free DNA fragments. The transcription start site coverage feature is based on the biological mechanisms of when a gene is active, its transcription start site is open and not protected by nucleosomes. Therefore, it can be completely degraded, resulting in a coverage dip in this active site. GDM is characterized by 3 core pathophysiology, including insulin-resistant B cell strain, placental endocrine dysfunction, and low-grade inflammation. These conditions can potentially be detected via the following multi-omic signals: total cell-free DNA load, fragmentomic shift, methylation maps, and placental cell-free DNA. Preterm birth is a syndrome of premature parturition activations, and it's there are multiple distinct pathological processes which are summarized here that is indicated in preterm birth. These conditions can be reflected by a multiple multi-omic signal, including cell-free DNA fragmentomics, cell-free DNA RNA, and proteomics and lipidomics. So GDM and preterm birth represent 2 common complications. Approximately 14% of pregnancy are affected by GDM, while about 11% of live birth are pretermed. Complications arising from preterm birth are the leading cause of death globally among children under the 5 years of age. However, current screening for both preterm birth and GDM are limited. In GDM, oral glucose tolerance test between the weeks 24 to 28 is late, and it misses the critical window for interventions. The current standard of care for preterm birth is also late with modest sensitivity, and most spontaneous preterm birth occurs in women with no identifiable risk factor. So our challenge is to use the same NIPT data, which already capture the fragmentomics of cell-free DNA, to make two new predictions for GDM and preterm birth. A literature review shows that this is highly possible with multiple studies achieving the promising result. However, not all studies can achieve good prediction with NIPT data, with its shallow sequencing data only. So in the remaining slides of this talk, I will share with you our approach and result, aiming to use NIPT data to predict both GDM and preterm birth. So we first look at GDM. In this study, we recruit 433 GDM pregnant women and 651 control women. So we collect blood at weeks 9 to 12, exactly as we do for NIPT, and we confirm the GDM as week 24 to 28 as per standard. For the data from NIPT test, we look at two different types of features, the fragmentomics features and the genomics feature. So for fragmentomics, we calculate multiple different types of fragmentomic features, some of which I have talked to you about before in my slide. For the genomic feature, we basically use the shallow sequencing data to impute the SNP. And with that, we can arrive at a score that represent GDM genetic risk for the patients. So we train two models, one for each of the type of features here. And for each model, we arrive at multiple different scores represent the risk of GDM from the features. And we optimize the score and pick the best score to combine together in the second stage to arrive at a master score for GDM prediction. In this rock curve plot here, you can see that there's a lot of different curves around this area coming from multiple different scores from either the fragmentomics or the genomic traits. But by combining the two together, we can see there's a big shift in the AUC jumping to 87%. This demonstrates that the two scores that we use are complementary. We also show that there's a way to use this master risk score for GDM for risk stratifications. So in a population of women scoring 0.8 and above, the GDM prevalence in this population is exceeding 70%. So these stratifications help the physicians identify a group of patients that can benefit from early interventions. In the preterm birth study, we recruit 286 participants, and we split them into training and validation cohort. With the training, we use the data to extract the feature for machine learning model training, and we also compare multiple different models to pick the best one. For the wet lab protocol, it is staying exactly the same as the NIPT protocol. The only difference is the bioinformatics, where we extract different multiomics feature, fragmentomic features, and do the predictions. So a quick look at the fragmentomic features that help distinguish preterm birth and control. So basically, we have two cohorts, and we plot the fragmentomics feature of both cohorts side by side. And initially, it's somewhat disappointing to see that there's not much difference between the preterm birth and the control groups. The biggest group and features that have the difference is the end motif in panel A and B here. So regardless, we actually build multiple different models, each for one fragmentomics features. So in here, I show you 5 different model from 5 different fragmentomics features. And out of these 5 model, you can see that end motif is by far the best performance model. And with this end motif model, we can achieve a sensitivity of 94% at the specificity of 93%. So this is very encouraging. So in conclusions, by using one blood draw and with no additional sequencing cost, we have developed 2 new AI models to predict GDN and preterm birth at weeks 9 to 12. So the added value for 2 new predictions at no additional wet lab cost is a major win for pregnancy care. However, we are still in the process of doing clinical validation to prove the clinical utility of these 2 test predictions. And hopefully with a positive result, Gene Solution will make this a reality in the near future. Thank you very much for listening, and Dr. Han, I will take your question. Thank you very much.
Thank you to our distinguished speakers for their insightful presentations. We'll now move on to a panel discussion and Q&A. But before we open the floor for broader discussion and questions from the audience, allow me to kickstart the discussion with some focused questions for our speakers. Dr. Natalie Chandler, if I may begin with you. What lessons from the NHS NIPD service for high-risk monogenic disorders are most relevant when considering broader responsible implementation of non-invasive testing beyond aneuploidy?
Good question. I guess we've learned a lot about the importance of having confidence in every single variant we're testing for, and that we can actually detect it. And we get really, for us, it's really important we get the depth required, and we have changed our internal quality control as we've learned from the service. And I feel that's really important. And we only do that by collecting our outcomes and knowing what the results are, and then we can make sure that our service is improving. I think external quality assurance is actually really, really important, because it's not just in, are you getting the correct result, but it's how you're reporting it to the patients, and that's assessed as well, and what information you're providing on the technique. And I think that's something that could really, we really need to develop in this area.
Yeah, no, thank you. Thank you very much. So may I move on to Dr. Sang Hung Tang? So what were the most clinically significant findings from your analysis of over 225,000 Asian pregnant women based on your extensive experience from implementing integrated NIPT in clinical practice? What were the key learnings? Thank you.
Yeah, thank you for the question. So the lesson learned from our study, we can see that our approach to utilizing the NIPT sample and integrate the carrier screening for the mother will improve the clinical implementation of the carriers. And our real-world data actually show that it's very effective intervention at the population scale and very cost-effective. But the lesson we also learn from our real-world data that we need to we go and advocate for the ACMG principle, that we on of the lab, on of the test for the carrier screening, we should follow the guidance of the ACMG Tier 3 to pick up the CB and the common disease. But the principle should be tailored to the genomic data of the population. For example, our data show that maybe cystic fibrosis may be very common in US and Europe. Now we have the evidence that it may be common in Asia population. On the other hand, the Prader-Willi syndrome is in the recommendation of the ACMG because it's been disease and very severe. But based on our study of the Vietnamese population, it's extremely rare. So that we need to combine the principle based on the tailored genomic data, and that we hope that we can get to the universal carrier screening with a panel gene that maximize the clinical impact for any country or any population. We apply the carrier screening and the NIPT. Thank you.
Thank you very much for your answer. So let me move on to Dr. Hua with Dr. Min Wee Van. TriSure ProCare enables early gestational diabetes and preterm birth risk assessment from the same routine NIPT blood draw. What clinical value do you see this integrated approach delivering?
Thank you, Ida, for the question. I think for us, as some of the scientists in the biotech industry, I think the most significant clinical value we see with this implementing is that we will shifting the paradigm, the timeline of intervention from reactive to proactive. So both of the GTM and preterm birth are also on cause from the early placenta development disorder. And usually standard diagnostic catch them too late. So we want to move that into early stage so that we can help some of the early management to prevent that from happen or help better outcome from the delivery. It also signals that how we able to do workflow efficiencies as well as to patient compliance in terms of in practice in emerging market, because in those country pregnancy already involved a heavy burden of clinical appointment and blood draw. So we want to consolidate into a single drive and then trying to stratify the patient, the patient into high risk versus low risk and help better management for that. Yeah. Thank you.
Thank you very much. So now we'll open the discussion to all panelists. So with how do we come up with a consensus of what should be included in non-invasive prenatal screening for monogenetic disorder panels? So who wants to start?
Shall I start? I guess we need to consider what conditions the test has been shown to be analytically valid for, what the questions we're actually answering. So it depends on who's referring in. Then I guess the severity of the condition, and that might vary in different countries, and so we need to come up with some consensus of that. And then I think treatable things we should be looking at for things where fetal therapies are coming up. But yeah, how we come up with some guidelines. I know there's some papers on trying to find genes in relation to fetal therapies. There have been some papers on that, but I think some discussion needs to be considered on the like the de novo dominant conditions and what we should be screening for there. And I guess like for carrier screening, there are already the guidelines that are out there from ACOG or ACMG. So I'm open for others' views as well.
Dr. Sang, do you have any comments?
Yeah, thank you, Ida. So just follow Dr. Chandler comment. So and I already mentioned that actually we need to follow the principle of the prenatal screening that focus on the common and severe disease. For the recessive disease, actually we have the ACMG guideline. But for the dominant disease, actually we do not have the consensus and also the genomic data. So to screen for the most common and severe disease tailored to each population. So I think like that we but when we implement into the real world and combine the prenatal screening with the prenatal diagnosis and also postnatal diagnosis for each country, we start to see emerging signal of the important disease that we should not forget to include into the panel. And of course, we believe that the genomic study will open the new era for us and also get us on how to tailor the panel gene to maximize the clinical impact for each patient and also each country.
Thank you. Thank you very much. So maybe if nobody else have any question or want to add on to Dr. Natalie and Dr. Sang, maybe I'll move on to the next question. So with early risk stratification now possible for DDM and preterm birth through integrated NIPT platforms such as the TriSure ProCare soon, what proven effective early interventions exist and what evidence on treatment or intervention should we gather to fully capitalize on this opportunity? Maybe Dr. Hua, you want to go first for this one?
Thank you, Ida, for the question. I think I can speak first for GDM. I think for GDM, with the early prediction, risk prediction from NIPT data, I think the next step for the mother definitely will need to take the OGTT test at 24 to 28 weeks to confirm that for sure. But during that time point from the first trimester to the second trimester, I think the mother can manage their diet and then control through a lot of and do a lot of blood tests to manage that process throughout the time to make sure that if they don't develop don't develop the GDM at about 28 weeks, I think that's a good management intervention that we can start early. Other than that, I believe there are many monitoring that we can do with the patient throughout if it actually develops GDM at that phase. Yeah.
Thank you. Thank you. Dr. Hui, you want to add to that or as well? Or
I think Dr. Hua has spoken for me.
Yeah. Okay. Dr. Chandler, do you want to add to the question as well?
I think there needs to be a huge
Comments?
amount of research done here, and we need, you know, massive studies on alongside the therapies and kind of the use of this technology and the health economics, the social science, the patient input. I think we need to have some really huge studies done in this area, and I think, you know, it's a really exciting area, and I think these studies are going to happen, and it's going to be great to see how this happens.
Thank you very much. So I think maybe time for one last question before closing. So maybe why is chromosomal placental mosaicism being not an issue for SMVs like it is for aneuploidies and some SMVs?
Shall I start? So there have been studies showing that you do get placental mosaicism for SMVs as well, and they form these kind of placental clones. But obviously, unlike aneuploidies, where its whole chromosome changes, there's millions of bases that can be changed. So the chances of one of those being a pathogenic variant on your panel is actually really low. But equally, the studies have also shown that when they have occurred in the placenta, they're not showing up in the cell-free DNA. So even though it has been reported you can get placenta mosaicism, the data so far suggests that it won't be present in the cell-free DNA.
Dr. Sang, you want to add to that?
Yeah, thank you, Dan. So actually, based on our experience, we also experience up the confined placenta mosaicism for the aneuploidy and the copy number variation, but we do not experience for the monogenic disorder. But we think like we need more study to confirm that, and also more study in the future to confirm whether we have the CBM for the monogenic disorder or not. Yeah.
Thank you very much. So on behalf of Gene Solutions, I would like to express my sincere appreciation to all our distinguished speakers today for sharing their expertise so generously. This symposium has underscored both the robust evidence of supporting prenatal non-invasive prenatal testing and the promising opportunities that lies ahead, particularly through platforms such as the TriSure ProCare. In addition to this industry symposium, Gene Solutions and the Medical Genetics Institute of Ho Chi Minh City are proud to contribute further to the ISPD 2026 through 5 scientific abstracts, including 2 oral presentations and 3 virtual posters. We will also have another scientific session at the same time tomorrow. Thank you once again for joining us, and we look forward to continued collaboration in advancing prenatal genomics. Thank you very much.